I came across with this bungle plant upon strolling around our neighborhood. Does it look familiar to you? It pretty looks like SARS-CoV-2, which causes COVID-19, right? But how do we know that SARS-CoV-2 really looks like this? And haven't you wondered how swiftly COVID-19 vaccines were produced? Well, let's get to know structural biology and the amazing imaging technique used for it. Structural biology aims to understand how biological molecules such as proteins are built. Like if this is a protein, structural biologists use imaging techniques to see how these proteins are assembled, how they function, and how they interact. Cryo-electron microscopy, or Cryo-EM, is the newest imaging technique used by structural biologists. Traditionally, there are other techniques, but they have several limitations. In October 2017, three scientists shared the Nobel Prize in Chemistry for their work in developing Cryo-EM. With Cryo-EM, biomolecules such as proteins are swiftly frozen. It's like being cast with the Petrificus tablets, or body freezing spell by Horror Watson. Using an electron microscope, the frozen sample is fired with a beam of electrons at the speed of light. Emerging scattered electrons pass through a lens and forms two-dimensional and amplified image that is recorded on a detector. A computer algorithm combines and sorts images taken from several angles, and from that, a 3D model of the biomolecule structure can be generated. Cryo-EM is very crucial in our fight against COVID-19. Using Cryo-EM, the collection of about 3,000 images to map out the spike protein of SARS-CoV-2 only took 24 hours. A decade ago, just creating these images could have taken weeks or months. With Cryo-EM, in less than 2 months since the coronavirus was reported, scientists already had significant understanding about the protein structure and the behavior of the virus. Such progress could have taken years in the past. The accelerated process of uncovering the protein structure of SARS-CoV-2 paved the way for the speedy development of vaccines such as that of Pfizer-BioNTech's and Moderna's. Indeed, the advances made by Cryo-EM and structural biology gave us tremendous advantage in understanding emerging and infectious disease, and its possible applications in vaccine development and therapeutics is just limitless.